All right, folks, this is a short video on meiosis and genetic variation. Okay. In this video, I'm mainly going to be focusing on three things. One is going to be independent assortment. Okay. I'm also going to focus on how to determine the possible combinations of chromosomes due to independent assortment. And then I'm also going to talk about crossing over, okay, both of which occur during meiosis 1, and you're going to have an increase of genetic variation because of those two processes. Okay, so the first thing that I want to show you is a picture of independent assortment. Like I said, it is it looks like a complicated diagram, but it really isn't. Okay, so here I have my chromosomes. Okay, so I have my homologous pairs here, right? Now, when you have 4 of them like this, in addition to calling them homologous chromosomes, right, and you can call the 2 blue ones, you can call them sister chromatids, and you can call the 2 red ones sister chromatids. And then we have if you look at the blue and the red one, those would be called non-sister chromatids. But we really haven't assigned a name for all 4 of them together. And one word that we tend to use a lot in genetics is the word tetrad, right? Like tetrahedral, meaning 4. So when you have all 4 chromatids collectively, right, these 4 chromatids, these 4 homologous chromosomes, we can call them a tetrad. So here during this is happening during metaphase 1, so this is happening during meiosis 1. Okay, what we can see is 2 possible, right? This is the first possible arrangement. This is the second possible arrangement, right? The first possible arrangement is along this equator that runs north-south of this cell, right? The blue chromosomes are showing up on the left-hand side, and the red ones are showing up on the right. So once anaphase 1 occurs and we have the separation of the 2, and then it finishes and goes through the rest of meiosis 1 and then starts meiosis 2, when we get to metaphase 2, if we notice that this homologous chromosome right here and this homologous chromosome right here, these 2 right here, my bad, they're from the same source, right? They both came from the blue parent, right? These 2 both came from the red parent. We still have the same the correct division going on, or the correct segregation of the chromosomes, right? There's 2 chromosomes. We have a big chromosome and a little chromosome. And after meiosis 1 split and the tetrads get pulled apart, right? And then we have these homologous chromosomes in 2 separate cells, blue and red. Now we can see that these 2 chromosomes came from the blue parent. These 2 came from the red parent. So when meiosis 2 finishes and you end up with 1 chromatid in each cell, right, you end up having only, this possibility or that possibility there, and then this possibility there, this possibility there. Obviously, they're the same thing, okay? But what's just as likely to happen is in the chromosomal arrangement 2, which is the bottom half of this screen, where it's completely random, where the homologous chromosomes here are ordered, or the tetrad in here is ordered blue to red, and then down here for the smaller chromosome, it's red to blue. So when meiosis splits them, then you end up with a big blue and a little red over on this cell, and then a big red and a little blue over on this cell. Okay, so when they split, yeah, they're going to be genetically identical here, and they're genetically identical there. But if you notice that just based on these 2 chromosomes, right, chromosome blue and chromosome red, right, for these two, and then these 2, there is 4 possible combinations. We could have blue-blue, we could have red-red, we can have big red, little blue, and we can have big blue, little red. So the variability in these 4 gametes really comes down to the random assortment during metaphase 1, right? When these tetrads are lining up along the equator, it could be blue-red, like it is up here, or it can be blue-red, red-blue like it is down here. And so when they split, then we could have, big chromosome is blue, little chromosome is red. But over here, it could be little chromosome is blue and big chromosome is red. Up here, they kind of leave together. They're coming from the same sources, right? Now, if you're looking here, how we're identifying diploid and haploid, right? This is an important thing to note, right? Count the number of centromeres here, right? That's the connections, right? Well, how many connections do we have here per cell? We have 1, 2 for each chromosome, right? So there's 1 and there's 2. 1, 2. So this is a diploid cell. After meiosis 1, there's only 1 per chromosome. So there's only 1 centromere per chromosome. So even though there's 2 copies of the genetic material in here, it is still considered a haploid cell because they're nothing but duplicates of that same chromosome. So here you have diploid because it's coming from 2 different sources, right? And you can just honestly just count the centromeres, count the connections that are holding these chromosomes together. There's 1 connection here and 1 connection here for the big chromosome, diploid. So here's 1 connection here, but there's only 1, haploid. If I look down here for this chromosome, there's only 1 connection, haploid. So everything after meiosis 1 are all haploid cells, even though there's essentially double the amount of genetic material. But it's, like I said, it's just a copy of that same piece. So looking at, this is a good video, or a good illustration here to show the difference between haploid and diploid. Okay. Now the next question is, well, how can I determine the total number of possible combinations for our our expectations? What are the possible combinations for these 2 chromosomes, right? We calculated it, or we actually determined there's 4, right? There's this one, this one, this one, and this one, right? How can I show that mathematically? What if I had, 5 chromosomes, not 2? Or if I had 23 chromosomes like we do, right? Well, there's a way to mathematically do that, and it looks like this. So this is from our example. Okay, the formula is going to be 2 to the n. Remember, n is essentially our haploid number. So when we're looking here and we're trying to find what our haploid number is, right? If I come back over here, I have how many chromatids do we have, or how many chromosomes do we have here in our haploid cell? Well, there's 2. There's this chromosome here and this chromosome here. So that's our haploid cell. Our haploid cell number is 2. So in this case, we had at the beginning, our diploid number was 4. And then when it split after meiosis 1, our haploid number, n, was 2. All I got to do is put the 2 where that superscript is to go 2 squared equals 4. Okay? And how I could calculate that is I'd go, this is just a calculator I pulled offline right here. I'd go 2, and then you might have a caret key, which is like a little arrow, but looks like it's pointing up without the little leg, just like those, see if I can find a nice little, it looks kind of like this, but it's pointing up, not pointing to the side, right? So if you look over here, that's essentially what a caret key looks like. I don't think I have one on my keyboard here, but if you have a calculator like this, it's just the X to the Y button. So let's say I had a haploid number of 2. So I'm going to go 2 to the second, and that'll give me 4. Well, what if I had a haploid number of 15? So 2 to the 15. And then the total possible combinations, right? When we were looking at this, there were 4 possible combinations here. But now when I look at it, it would say if I had 15 different chromosomes, just randomly, independently assorted, I'd have 32,768 possible combinations. Okay. Now, the reason why I'm telling you this is because you have a couple evidence notebook questions. Okay. This is the evidence notebook question that is on page 269. 269. This is the problem-solving questions. This is what needs to go in your evidence notebook. To determine the number of possible chromosome combinations, the result from independent assortment, you can use the formula combinations equals 2 to the n. We just showed you that. So where n equals the number of chromosomes, or in this case, the haploid number. What is the approximate number of possible chromosome combinations for a human cell with 23 different chromosomes? So you're going to need to do the same thing here. You'll go 2, and then for your exponent, you're going to go 23. I'm not going to type it in because it's going to give you the answer, and we don't want to do that. Okay, so you would go 2, and then do your caret key or your XY button, and then put in 23, and it'll give you a value, and that's the value that you would put in for your audio question. Okay. Don't forget, you also have an evidence notebook question here. Feel free to pause this video so you can write these questions down. All right. The next thing I want to talk about is crossing over. So crossing over is when you have, this happens during prophase 1. Okay, this happens during prophase 1 when you have your tetrad. So here's my tetrad. And then within the tetrad, I have my 2 sister chromatids for one parent source. And I have another 2 sister chromatids from my other parent source. Okay, so here I have one chromatid here that are not from the same parental source. We would call those non-sister chromatids. Crossing over is when during prophase 1, 2 non-sister chromatids swap sections. Okay, just because they're, DNA, we can, it's the building block and it's the instructions for everything, but it's also fairly fragile when it comes to the ability to break and then re-break. So as it turns out, during meiosis 1, specifically prophase 1, we'll have a section of different chromosomes break and then switch on non-sister chromatids. It is possible to end up having it, break and then re-break and then break again and then re-break again and reform, right? But important term here, recombinant chromatids are the non-sister chromatids where crossing over took place. Non-recombinant chromatids are going to be non-recombinant chromatids are the 2 non-sister chromatids that are not that did not do crossing over. Okay, so obviously, if I was to reintroduce this picture, and then if we had some swapping going on here, then it really increases the chance of having genetic variation happening in the chromatids, right? By the time you get to the gamete stage, there could be a wide variety of different breaks, considering that the number of breaks that occur in meiosis, or sorry, in crossing over is not determined by anything. It's random, right? So independent assortment, crossing over, big deal. Here is your evidence notebook question. That's going to be your end of section question. So feel free to pause the video and write it down and answer it in your notebook. If you have any questions, feel free to ask.